Chapter 7 covers bacterial and viral genetic systems. We won't be covering everything that's in Chapter 7, so stick close to your PowerPoint lecture and take good notes. We'll be looking at the main ways that bacteria can recombine their chromosomes and the way viruses can recombine their chromosomes, and a little bit about the history of HIV, for example. So the advantages of using bacteria and viruses are shown here. For the most part, they're really vast model organisms or models to study because they reproduce rapidly. You get a lot of progeny. They only have one copy. Generally, they only have one copy of the genome, and so you get to see the phenotype directly from the genotype. They don't take up much space. They're cheap. They're easy to manipulate. And they have medical importance, and they have really a lot of importance of commercial value of all types, including our agricultural applications, pharmaceutical applications, and so on. So the way that you grow bacteria, let's talk about bacteria first. Bacteria can be grown in a liquid medium that's called broth, or if you mix broth with something called agar, and it's like a white powder, and you boil it and let it cool, it makes a solid that you can pour that hot molten agar into a petri dish, and then it solidifies, and you can grow bacteria on the solid medium. If you grow it in liquid, it's called a culture. If you grow bacteria on a plate, it's still a culture, but it forms individual colonies. So prototrophic is a term that we use to refer to bacteria that are wild type. That means they're able to synthesize all their own biomolecules. The only thing that needs to be provided is an organic carbon source, which is a sugar. An auxotroph would be an organism that has a mutation in a gene that causes it to require some kind of biochemical supplement in order to be able to grow. Bacteria, when grown on a plate, like I said, form colonies. A colony grows from a single bacterial cell into a pile of clones of that bacterial cell. And interestingly, bacterial colonies have phenotypes of their own, including color of the colony, edge, puffiness, size, all sorts of different characteristics that bacteria can have when they grow in colonies, and some examples are shown here in pictures A and B. Some bacteria have extra genes or have mutations in genes that lead them to have resistance to antibiotics. So that is a mutation or a special characteristic of some bacteria. And some bacteria have restrictions for what type of carbon source, so what type of sugar they can utilize. As a rule, bacteria only have one chromosome and then smaller plasmids. So this is a micrograph showing a bacterial cell that's been broken open. You can see the chromosome, which if you stretched it all out would make one very large circle. And then these smaller circles called plasmids, you can definitely see those as well. And the number of plasmids, there's no set number. Some bacteria have lots of them, some don't have that many. And so but bacteria just have one chromosome per cell. Replication of that chromosome and of the plasmids takes place through the type of bidirectional replication that you learned in Biology 1406. There's an origin of replication, synthesis of the new strand, shown here as sort of the light green color, originates at the origin but then moves bidirectionally on both strands. As the replication forks move away from each other, you start to see under the microscope something that looks a little bit like the Greek letter theta. So this is called theta replication, and by the end of the process, the replication forks eventually collide at the bottom of the molecule, and you end up with two separate chromosomes or two separate plasmids, two separate molecules. So what we're looking at first in Chapter 7 is the process by which bacteria can recombine their genetic molecules, because bacteria don't have meiosis, they don't have sexual reproduction, but there are ways that bacteria can exchange genetic information. And these ways of exchanging genetic information are sometimes called horizontal gene transfer. But what happens is there's three processes that can lead to this. One's called conjugation, one's called transformation, and one's called transduction. But what's important in all of these is that even though there's three different ways that genetic exchange can occur between bacterial cells, the end product in each of these is basically the same. So they all have the same outcome. So you can see here in this picture there's going to be a bacterium that's considered the donor. It's going to donate some genetic material. There's a cell that's going to be the recipient. It's going to recombine some of the donor's genetic material. And they're using this color coding, the red chromosome from the donor, the green chromosome for the recipient. And you can see at the end of each of these three processes, you end up with a recipient that has incorporated a piece of the donor's chromosome into its chromosome. And so that's, a recombination within a bacterial cell, or between two bacterial cells in a sense. So that's how there is genetic exchange in bacteria. And so we'll define each of these processes, but remember they each really end with the same product, a recombinant bacterial cell. The historical experiment that corresponds to our discussion of recombination in bacteria takes us to what's called the Lederberg-Tatum experiment. And that's in 1946, so this is really only 2 years after Avery, McLeod, and McCarty. And they asked the question, do bacteria recombine their chromosomes? Because they don't do meiosis, they don't have sexual reproduction. So the question here at the top of this, do bacteria exchange genetic information? And you can see down here the conclusion is going to be yes. But the experiment they did, they had two strains of the same species of bacteria, but the bacteria had different genotypes. And so you're seeing a close-up here. The blue chromosome is one, that's strain Y10. That represents the chromosome in each of those cells. And then strain Y24 has a different genotype for the same for those same genes. And so you can see if there's a plus, that's normal allele, and if it's a minus, that's a mutant allele. So for example, strain Y10 is 3 minus, Lu minus, Thi minus, Bio plus, Phi plus, Cysteine plus. So these are all abbreviations for molecules that the gene products would allow the cell to make. And so strain Y10 we could say is an auxotroph for threonine, leucine, and thiamine because the genes necessary to make those biomolecules are non-functional. But the cells are able to make biotin, phenylalanine, and cysteine. Whereas strain Y24, you have its normal alleles for threonine, leucine, and thiamine, but mutant for biotin, phenylalanine, and cysteine. So separately, when they plated these cells on minimal media, which just has the basic sugar source, no extra supplements, neither strain can grow. So that's shown here. But when Lederberg-Tatum then mixed the two strains of bacteria together into the same test tube and then plated that out, they didn't get a lot of growth, but they got some colonies. And when they studied those colonies biochemically, they found that those colonies were completely prototrophic. They had normal alleles for all the genes. And that indicates that somehow the two chromosomes from the two different bacteria had been able to recombine. They didn't know how that happened yet, but they answered their question. Yes, there's some kind of genetic exchange that's possible. Maybe not real common, but it happened at least a small number of cells. And this picture just shows somehow there was a recombination between the two chromosomes of the two strains, and you can see that it's possible to get a strain that's completely wild type. We call it prototrophic, which would be a recombinant of the two. So some cells were able to get this combination of wild type alleles. Then Lederberg-Tatum, their experiment was taken further, and they put their two strains into a U-tube, which is separated by this filter in the middle, and the filter has holes in it, but only liquid is able to cross, not cells. So the cells are not able to cross through the pores in the filter. And when they restricted the movement of the cells, when the cells weren't able to touch, so the Y10 and the Y24 cells were not able to touch directly, then nowhere in the experiment were they able to recover any recombinant prototrophic bacteria. So that led them to the conclusion that the type of genetic exchange that they were observing would require direct contact between the two cells. It turns out that Lederberg and Tatum were observing one of the types of recombination that exists with bacteria, and that's a process called conjugation. And conjugation involves the transfer of a genetic molecule called an F factor from one bacterial cell to another bacterial cell. So you'll have a donor cell that has the F factor, you'll have a recipient cell that doesn't have the F factor, and the transfer will happen in one direction. This shows a typical F factor. It has an origin of replication, and it also, right here, sorry, and it has genes that allow it to make what's called the pilus, which creates a bridge between the two cells, and then some other proteins that allow for its replication and transfer into the other cell. This picture shows a donor cell which has these pili. These are extensions of the plasma membrane constructed because of the genes on the F factor. And one of the pili, one pilus, will extend and connect to a nearby cell, and the cytoplasm is shown in red, and you can see that it creates a continuous connection between the cytoplasms of these two cells. And then the F factor, that DNA molecule, will travel across this bridge, this pilus, and go into the recipient cell. And this is showing that in a more schematic way. You have the F factor in red. Here's the pilus shown here. Now, the cell that has the F factor is called F plus. A cell that doesn't have it is called F minus. So the F factor will be transferred to the recipient cell, but only one strand of the F factor is transferred. And as it's transferred, it's replicated at the same time, and you end up with two F plus cells as a result. So the pilus eventually will break, but typically it will stay intact long enough for the transfer to happen. So that's not that interesting, except if you get some kind of gene on the F factor, which sometimes happens. Somehow maybe an antibiotic resistance gene gets onto an F factor, and then it can spread through a bacterial culture very quickly because it's being transferred with all of the other genes on the F factor. All right, now, also part of conjugation, the F factor has a special characteristic. It can integrate onto the host cell's bacterial chromosome, and in that case it creates an HFR cell. So if you have an F factor that's independent like this, but then it has a single crossover with the larger chromosome, then you have a cell called HFR. HFR is like F plus, except HFR, that indicates that the F factor has actually connected directly with the chromosome. And just like with an F plus cell, an HFR cell will conjugate with an F minus cell. However, the transfer of the entire chromosome takes a lot longer than the transfer of the F plasmid by itself. And I asked the question, and it's true, it takes about 2 minutes to transfer an F plasmid to an F minus cell. How long would it take to transfer the HFR chromosome? Well, the HFR chromosome is 50 times longer, then it would take 50 times longer. Then if it takes 2 minutes, 50 times 2 is 100 minutes. And what happens is the F factor will initiate transfer, and it will actually pull along with it some of the donor cell's chromosome. But after usually 20 to 30 minutes, the pilus will break. It just doesn't stay intact for 100 minutes. And so what's typical with an HFR conjugation with an F- cell is that you only get a portion of the HFR chromosome transferred. Now, a linear piece of DNA that enters a bacterial cell will typically get destroyed by restriction enzymes. However, and so most of the time, this piece of DNA here in part D will just get destroyed and you'll have no effect. You won't see anything happen. But it does happen rarely, but it's possible to get the section of the donor chromosome to integrate onto the recipient chromosome, and you can have a recipient that changes genotype for a gene. Now the cell stays F- because the entire F plasmid doesn't get transferred. The red section is not completely transferred, but a different gene is transferred, and so this F- cell might have a new genotype. So for example, if this black allele was Lu- and this green one is Lu+, then the F- cell, which is Lu-, until it recombines and it becomes Lu+. So it's still an F- cell, but it might change genotype for one of its genes if that crossing over event happens.